BISC130, this is the beginning of recorded lecture 1.3. We're still in chapter 2, Chemical Foundation of Life. And, we talked about water last time, and there's one more thing to talk about that is related to water, the pH scale. So the pH scale ranges from, it's a scale that ranges from 0 to 14 with the definition of the middle here, the definition of 7 being pure water. So what is the scale actually measuring, though? Well, let's take a look at this. I don't expect you to memorize all these examples here, but this is just a visual for this. 0, 14, and there's, distilled water or pure water right here at 7. And we've got examples of things that have lower pH values, examples of things that have higher pH values. So what is pH actually measuring? Well, if you'll remember the previous lecture, one of the properties of water is that water ionizes. It splits apart into OH minus hydroxide and H plus hydrogen ions. So pure water has a certain amount of hydrogen ions in it, but other solutions may have more or fewer of these hydrogen ions, and that's what pH is measuring, the concentration of these hydrogen ions. So the pH scale related to concentration of hydrogen ions. Importantly, and somewhat confusingly, this is an inverse relationship. So to go back to this scale, a lower pH value means a higher concentration of hydrogen ions. Something like tomato juice has a lot of hydrogen ions compared to distilled water. Something like the milk of magnesia has a higher pH value. It's 10 instead of 7. It has a much lower concentration of hydrogen ions compared to the middle here, distilled water. So that's what I mean by an inverse relationship. And just to put it in even more clear terms, if you have more hydrogen ions, you are more acidic, which means your pH is lower. If the solution has fewer hydrogen ions, that means it's more basic, and the pH is higher. Higher pH, fewer H+, lower pH, more H+. The other interesting thing about this scale is the relationship between these values. So, 7 to 6, like urine here. So we know urine has more hydrogen ions than distilled water. But, how many more? Is it like, one extra hydrogen ion? Is it double? What is it? Well, this pH scale is a logarithmic scale. And if you're a math person, you know exactly what this means. If you're not a math person, don't worry. I'm never going to make you do calculations or anything on my exams that use this complicated math. The short version is log scale means it's a tenfold scale. There are tenfold differences between whole numbers within the scale. So again, just to list an example, that pH 6 has 10 times more hydrogen ions than pH 7. We can go back here and do a couple more examples. If we wanted to compare tomato juice to distilled water, that is tenfold to go here, another tenfold to go here, and another tenfold to go down to 4. So 10 times 10 times 10 is 1,000. Yeah, you may have to do math like that on an exam, but 10 times 10 is something you should be able to do in your head. And we could go the other direction. So let's compare distilled water to seawater. Going from 7 to 8, that is 10 times fewer hydrogen ions. Or if we want to go up to a baking soda solution, 10 times, 100 times fewer hydrogen ions. That's all you really need to know, that there are tenfold differences between these whole numbers. So there are a couple of other things related to the pH scale. I already referenced, being acidic and basic, referring to things with a lower pH being called acidic and solutions with a higher pH being called basic. But acids and bases are actually in the key terms. So things that we call acids are things that, when added to a solution, will lower the pH. The key terms define an acid as a molecule that donates hydrogen ions and increases the concentration of hydrogen ions in a solution, which makes sense. How do you lower the pH? You put more hydrogen ions in. Let's take a look at this in action. Here is carbonic acid, an acid, obviously. And here we see its molecular structure. Don't memorize this. But yeah, you can see a little hydrogen up here that pops right off and is put into the solution. Putting carbonic acid in a solution releases hydrogen ions. That's going to lower the pH. That makes this an acid. In contrast, bases do the exact opposite. A base increases the pH and it removes hydrogen ions. Let's say, how do the key terms phrase that? A base is a molecule that donates hydroxide ions or otherwise binds excess hydrogen ions and decreases the concentration of hydrogen ions in a solution. So a base is not going to make these hydrogen ions just magically disappear. Usually, they're going to bind them to themselves. So let's take a look at a base. This is the bicarbonate base. The bicarbonate removes hydrogen ions from the solution by attaching it to itself, and removing hydrogen ions is going to increase the pH. Now, you may have noticed that bicarbonate and carbonic acid are kind of two sides of the same coin. It's just this arrow pointing in different directions. Here it is acting like an acid. Here it is acting like a base. This illustrates a third key term in this section, and that is the term buffer. A buffer is a chemical compound that is going to resist change in pH. The key terms more formally say a buffer is a substance that prevents a change in pH by absorbing or releasing hydrogen or hydroxide ions. It could go either way. It could do either one depending on the circumstances. pH or buffers keep pH in a specific range. Again, they act as acids if the pH is getting too high. They act as bases if the pH is getting too low. They do both. This is a very important part of, to call back to a key term from Chapter 1, Lecture 1.1, homeostasis. We want to maintain homeostasis of pH in our cells and in the fluid around our tissues. Lower high pH can be bad for cells. So yeah, we want to make sure it doesn't get too high or too low. Buffers will do that. And yeah, here's an example of what this can look like. The carbonic acid bicarbonate buffer. It's just our two examples from before, but this molecule can go in either direction, preventing things from getting too high and too low. There are a ton of buffers that exist, another table that you definitely don't need to memorize, but some buffers will keep the pH at a relatively low range. Some buffers will keep things in a pretty high pH. Again, there are tons more of these that exist, but every single buffer has their special little range where if it gets too low, they'll start acting like a base. Too high, they'll start acting like an acid. Your take home from this colorful table is that there are many different buffers that exist, and they're active in living things. Again, part of the homeostasis of our cells and around our cells. Now, to finish out this chapter 2, there is one more topic here, and that is a topic about carbon. So in this whole periodic table of the elements, we talked about sponge on the first day of class, the elements that are most common in living things, but the one that's probably most important for us is specifically carbon. Why carbon? Well, it's got 6 protons, 6 neutrons, 6 electrons by default. You remember from last class period or last recorded lecture that these atoms are trying to get to 2, 10, or 18 total electrons. Carbon has 6. That means it wants 4 more. That means it can have up to 4 partners. If you're trying to connect these things together as like building blocks or Tinker Toys or some sort of building toy, being able to form 4 connections with other things is really convenient. So that's what's so important about carbon. That's why we're called carbon-based life forms in science fiction. That's why, when in the search for extraterrestrial life, they look for carbon-based things. Carbon is an ideal element in the universe for building molecules because it can have up to 4 partners. It can form covalent bonds with up to 4 different atoms. And we'll see examples of these kinds of organic molecules in the coming chapters. And yeah, carbon's going to be kind of the backbone of all of this, with 4 different connection points to other things. Just very convenient. But carbon alone is not enough to make robust, complicated, interesting, cool organic molecules. Carbon is sort of the backbone of things. What we do is we take a backbone of carbon and then we throw on other fun things called functional groups. Here's another table that you will memorize if you take a chemistry class, but for intro bio, don't worry about committing all of this to memory. But yeah, you can see there's carbon here and then something else added onto it, a double bonded oxygen or a nitrogen or phosphorus or sulfur or more carbon or an OH. We've already seen OH groups as parts of many things, this polar covalent bond. So just in general, these things are called functional groups, and you put these on a backbone of carbon to give molecules their special properties. So the take home from that, functional groups give organic molecules specific chemical properties. We'll see carbon backbones, we'll see functional groups as we move into the next chapter, which incidentally is what we're going to move into. So sometimes these recorded lectures will start with one chapter and move on to another chapter. That's just how things work. So now we're moving on to chapter 3, biological macromolecules. Now, within this chapter, we'll talk about various molecules that you're familiar with, especially through nutrition, carbohydrates, fats, sugars, lipids, proteins, stuff like that. Let's start out with carbohydrates. So you can organize your notes however you want to. I'm double underlining carbohydrates because we're going to have several types of carbohydrates that sort of go underneath this heading. So yeah, we've got carbohydrates. These are macromolecules that are classified based on their size. So again, there are going to be several types of these. They're based on how big they are. Importantly, carbohydrates are all hydrophilic. They love to be in water. They dissolve in water. When we start looking at some of their structures, we're going to see the parts of them that are charged, that are water-loving, that make these things as a whole hydrophilic. So I promised several types. The first type up is monosaccharides. Mono means one, saccharide means sugar, so yes, we're starting with one. The key terms define a monosaccharide as a single unit or monomer of carbohydrates. Yeah, these are the simplest ones. And if we want to take a look at what these things look like, again, you don't have to memorize any of these exact structures, but we do see some of the things I've been referencing. Here's our backbone of carbon here in glyceraldehyde, carbon, carbon, carbon. And then we've got some functional groups. Yep, there are some OHs here. There's a double bonded O. Yeah, remember, these OH groups are polar covalent bonds. These are going to be charged. That's what makes these things hydrophilic. If we look at some other monosaccharides, here are some more complicated ones like glucose, and yeah, carbon backbone, OH, 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 double bonded O. Yeah, all this stuff is partially charged. All these things are polar covalent bonds. This glucose is going to love to be in water. So the way because these things have a backbone of carbon, usually when we describe how big something is, whether it's a sugar or just any organic molecule, we will talk about how many carbons it has as a way to describe how big it is. So yeah, the most common monosaccharides have either 3, 5, or 6 total carbons, and that's a convenient, quick way to talk about how big they are. So monosaccharides, usually 3, 5, or 6 carbons. The bigger ones, the 5 and 6 carbon monosaccharides, can actually form ring structures. So in this figure, we were just looking at their linear forms. What they can do is they can fold up and around, connect to themselves, and form these rings. So yep, 5, 6 carbons forming these ring structures. And yeah, you can see the double arrow line here. They just go back and forth in solution between this linear form and a ring form. The 3 carbon ones can't do this, but yeah, the bigger ones can form these ring structures, and that's what we will commonly see. You see these rings. That's an obvious monosaccharide. So we've talked about what they look like, but what do they actually do? Well, monosaccharides are commonly used for energy storage. Now, this is an interesting concept that we'll get more into in later chapters about energy and metabolism. But the short version of this is that breaking covalent bonds is something that releases energy. If you break apart bonds, that's going to release energy that you can use to do stuff. So just by having these covalent bonds holding together the atoms in this molecule, that makes this an energy storage molecule. And yeah, a lot of these things are built just to be broken down at a later date in order to get energy. A great example of this is fructose. And yep, here's our fructose example found in fruits. That's where it gets its name. And yeah, these things make these monosaccharides so that they can use them for energy later, either themselves or by the bird or animal they're trying to entice to eat these fruits. But yeah, it's meant to store this energy for later. Example of monosaccharide, fructose, stored in fruits. There's another function that monosaccharides can have other than just storing energy. Because these things are relatively small, at least compared to the other carbohydrates we will talk about, these things can move around fairly easily. Monosaccharides can also be used for energy transport. A good example of this in animals is the sugar glucose. This is transported in the bloodstream. Yep, there's a glucose test monitor monitoring the amount of glucose in the bloodstream. But yeah, we have glucose floating around in our bloodstream in order to supply all the different tissues of the body with the energy that they need. So this is an example of energy transport, glucose in the bloodstream. Now, we started with monosaccharides. If we want to get a little bit more complicated, still talking about carbohydrates, if we want to get a little more complicated, we're going to move from monosaccharides to disaccharides. Mono means one, di means two. So a disaccharide is very simply just two monosaccharides connected together. Two monosaccharides connected by a special type of covalent bond, and I'll show this in just a second, called a glycosidic bond. So here we go. Here is monosaccharide glucose, monosaccharide fructose. Here is the very important glycosidic bond connecting them together, and bam, now we have a disaccharide. This one is called sucrose. So what are these things used for? Well, disaccharides are not that different than monosaccharides, and they're used for pretty much the same stuff. Here we have some refined sucrose here, table sugar cubes, but these are made in nature by plants. They'll store their energy in the form of sucrose, and they'll transport energy throughout the plant in the form of sucrose. So to summarize, disaccharides can be used for energy storage and or for transport. Sucrose is a good example of a disaccharide. It is glucose plus fructose. It's stored and transported in plants. And to give one more example of a disaccharide, again, there are dozens of different monosaccharides and disaccharides. I'm only giving a couple examples of each. Another example of a disaccharide outside of plants, I want to give an animal example, is the disaccharide lactose. So, you've got galactose, a monosaccharide, glucose, a monosaccharide. There's the glycosidic bond forming this disaccharide called lactose. This is also used essentially for energy transport, transporting from parent to mother to offspring here in breast milk. If you're lactose intolerant, your body is incapable of breaking apart this glycosidic bond, and taking in some lactose is going to cause some digestive problems. And so we'll talk about enzymes later in this chapter. Sorry if you can't see the tiny little text here. But if you're lactose intolerant and you can't break apart this glycosidic bond, this is a pill with an enzyme that will do that for you. So anyway, just a fun little side note here. My summary: Lactose is another example of a disaccharide. It is glucose plus galactose transported to infants in milk. Okay, so we have monosaccharides. We have disaccharides. You might be tricked into thinking the next one would be trisaccharides, but nope, we're not going to do 3. We're going to skip all the way to poly, which just means many. So anything that is more than 2 monosaccharides, usually it's lots more than 2, falls under this category of polysaccharides. The key terms define this as simply a long chain of monosaccharides. So yeah, a bunch of these things together. A polysaccharide is a polymer. This is a very important key term because we're going to see this come up a lot. We're going to see several polymers throughout this chapter. A polymer is simply a big thing made by connecting a bunch of small things together. What do the key terms say? The key terms say a polymer is a chain of monomer residues linked by covalent bonds. In other words, anytime you're taking small individual units and connecting them together to forming bigger structures, we call that thing a polymer. And yeah, polysaccharide is just a polymer of monosaccharides. These things can be branched or unbranched. Let's jump in and take a look at one of these. So yeah, here's individual monosaccharide, 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 and yeah, here's a branch point. So sometimes they're just a straight long line. Sometimes they have a branch point. So yes, these can be branched or unbranched depending on which one we're talking about. And the question arises, what are they used for? Well, these things are too big to really be transported throughout an organism. So polysaccharides are mostly used for energy storage. So polysaccharides can be used for energy storage. A great example of this in the animal body is something called glycogen, which is what we've been looking at here, labeled up here, glycogen. Glycogen is a polymer of glucose, and it is branched. So yeah, if you look more closely at the individual monomers here, this is glucose, 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 glucose. There's our branch point, glucose, glucose. It's just a bunch of glucose. If we want to zoom out a bit, because these are very large macromolecules, it's going to look like this. There's a colorful rainbow protein in the middle here to help hold it all together. More on proteins later in this chapter. But yeah, you can see these long branched chains with many branch points here. The whole function of glycogen is, like we said, energy storage. This exists to be broken down at a later date. If you want a little more background here, this is something that we store in our liver. So if you are fasting, if you haven't eaten anything in a while, your blood glucose levels are going to start getting low. In order to make sure they don't get too low, another example of homeostasis, your body is going to start breaking down glycogen in the liver to release glucose back into the bloodstream in order to keep those levels normal. So this whole molecule is built just so it can be broken down when you need it. And the fact that it's branched makes sense. Because it's branched, look at all these points where you could rip off a glucose. You could rip off a glucose here, or 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 here. You see my point? If this were an unbranched molecule, if this were a straight line, there would only be 2 points where you could start breaking this thing down. So in this way, the structure fits the function. It's branched, which makes it really easy to break down when you need it, which suits its function as being energy storage. So yeah, glycogen, example of a polysaccharide that stores energy. Stored in the animal liver, broken down to replenish glucose in the bloodstream. There's my summary. Now, there's one more function that we see in polysaccharides that we did not see in monosaccharides and disaccharides. Because these are very long, very large molecules, some polysaccharides can actually be used for structural support. Nothing to do with storing or transporting energy at all. Some polysaccharides are built just to be tough, just to be strong. A very good example of this is the polysaccharide cellulose. This is a polymer of glucose that is unbranched. So here we go. There's cellulose, glucose, 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 and you'll notice there are no branch points here. It is one big long line of things. You take many strands of cellulose, you connect them together with some proteins and some other molecules, and you make a very tough sort of cord-like fiber that really gives strength to specifically plant cells and plant cell walls. So again, the structure fits the function. It's unbranched, so it can form these tight cables, these stacked lines of this to be as strong as possible. And yeah, these things are very difficult to break down. Cellulose, part of the plant cell wall, very hard to break down. In fact, this is so hard to break down. If you eat a bunch of leaves or grass, okay, that sounds crazy, but no, if you eat cabbage, if you eat spinach leaves, if you eat celery, those things are loaded up with cellulose, which is just glucose. So you would think there would be a ton of sugar in spinach, in celery, in lettuce, and cabbage. But if you check the nutrition facts on these plant things, you're going to see it's almost 0 calories. Even though there's a ton of sugar present in leaves and stems of plants, it's inaccessible to us. We don't get any energy out of eating lettuce and cabbage because we can't access it. We can't break down this polymer. It's so good at being tough that we can't get the glucose out of it. So that it's good for the plants because it is less incentive to eat them. There are obviously animals that make a living eating leaves and grass and stuff like that. They have special ways of dealing with this, but many animals, including ourselves, don't waste our time on these things for the most part because, we can't access this. Just sort of tangent example about how hard this is to break down. Okay, that's enough about carbohydrates. We did monosaccharides, disaccharides, polysaccharides. All those things fell into the category of carbohydrates. For our next topic in this chapter, let's move on to lipids. So lipids, in contrast to the carbohydrates we just talked about, lipids are connected primarily by non-polar covalent bonds. That means no OHs, no unequal electron sharing. These connections, these covalent bonds, are going to be shared neatly, nicely, not making any partial charges, which is going to make these molecules as a whole hydrophobic. These things don't want anything to do with water because they're not charged. We'll see these bonds in just a second. Most lipids have at least one fatty acid as part of their structure. So before we really get into lipids and talk about the different types of lipids, I have to define what a fatty acid is because we're going to see this pop up in most of these lipid types. So fatty acids come in 2 basic types. Fatty acids can either be saturated or unsaturated. So before I read the key terms, let's actually look at this. So this is a fatty acid. This is another fatty acid. And, to call back to what I just showed, non-polar covalent bonds here. Yeah, look at this. C, carbon, 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 and hydrogens, 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 hydrogens. All this stuff is non-polar covalent bonds. There are no charges in here at all. So it makes sense that this would not want to hang out in water. So we can see labeled here saturated fatty acid and labeled down here unsaturated fatty acid, and it's pretty clear to see the difference. The saturated fatty acid is straight. The unsaturated fatty acid is crooked. It is bent. What causes this bend? Well, what causes it is a double bond. So in a saturated fatty acid, all of the carbons are connected by single covalent bonds. Anytime you have a double covalent bond between the carbons, that causes a kink. That causes a bend in the chain. So saturated fatty acids, no double bonds, straight. Unsaturated, at least one, sometimes there are multiple, this example is just showing one, at least one double bond between the carbons, and so at least one bend in the chain. To read the key terms definitions, again, picture's worth 1,000 words, so I'll read the key terms. You can look those up, make note cards, whatever you want to do, but yeah, these are great visuals for saturated versus unsaturated. The saturated key terms say, a long-chain hydrocarbon with single covalent bonds in the carbon chain. Unsaturated, to read the key terms, a long-chain hydrocarbon that has one or more double bonds in the hydrocarbon chain. And, if you want some practical connection here, saturated fatty acids pack together tightly. You can imagine a whole bunch of these stacked on top of one another. Foods that have lots of saturated fatty acids tend to be solid at room temperature because these things pack together just so tightly. Butter, lard, those things have lots of saturated fatty acids. Unsaturated fatty acids don't pack together so tightly because of this bend. Foods that have lots of unsaturated fatty acids tend to be liquids at room temperature. Things like olive oil or vegetable oil. They just don't pack together so tightly because the structures are, not just a brick of carbons. There's some wasted space here in the bend. Now, there's another type of fatty acid here that you may have heard of if you're keyed into, nutrition at all, and these are trans fats or trans fatty acids. Trans fats are technically unsaturated fats. I'll show an image of this in just a second, but they're artificially created. Trans fats do not exist in nature. It's a food processing thing that can be done that creates an unsaturated fatty acid. Look at that double bond here between the carbons, but it's not bent. This is a straight fatty acid, but it has double bonds in it. So, these are only found in artificially human-created foods. They're like a saturated fatty acid in that they pack together solidly and tightly. Sometimes we want foods, with properties of a saturated fatty acid, but it's cheaper to get unsaturated fatty acids. So they'll process these things to have the properties they want, but in doing so, they have this double bond here. Why does this matter? Why do people talk about these? Again, I'm not going to write this down, but diets high in trans fats in specific are correlated to increased risk for heart disease and atherosclerosis. So these things are generally pretty bad for you, and they're not found in too many foods these days. But yes, if you're curious what that is, it is an artificially created unsaturated fatty acid that is still straight like a saturated fatty acid. And one more. We got saturated, unsaturated, trans. Another one that comes up in nutrition is omega fats, or sometimes omega-3 fatty acids, omega-369 fatty acids. You'll hear these if you're in the nutrition world at all. The 3, 6, and 9 refer to the exact position in this chain where the double bonds are. This is, if we compare it to this is an unsaturated fatty acid. It's got a double bond here. It's got a kink in the chain. What makes these special is this very specific location of the double bond, especially when there are 3 of them like this. Again, don't memorize this exact structure, but in these omega fats, they're positioned in a very specific way, and we need these things. Again, we're going to use these fatty acids to build lipids. There are certain lipids that we need to make that use this exact type of fatty acid with this exact type of double bond in this exact position. And so these are really important as part of our diet. We cannot build these from scratch ourselves, so we need to get them from lots of different fish, and eggs and other seafood, some vegetables like broccoli and spinach. We need these as part of our diet to build the stuff that we need. So all I'm writing is omega fats are a type of fatty acid that's required as part of our diet. So again, I talked about fatty acids because lots of lipids are going to use these things. So getting on to lipids, well, we could break these down into 4 major types. So let's number these, why not? When I do things in my lectures and I number things, I'm never going to ask a test question, what's the 3rd type of lipid? The numbers don't really matter. The order doesn't really matter for these things. I'm just counting them so we can count to 4. So yeah, lipid number 1 is fats or oils. These things are going to be 3 fatty acids attached to a glycerol backbone to form a triglyceride. Let's get a visual on that. Yep. Glycerol backbone plus 3 fatty acid chains. There's a triglyceride, or this is triglycerol, triglycerides, same thing. 3 fatty acids attached to a glycerol backbone. That's our fat, that's our oil. So this is the structure of the fat or the oil. What about the function? Well, fats and oils are used for energy storage, which might sound redundant. We talked about carbohydrates that are used for energy storage. Why do we need another thing for this? Well, triglycerides are really, really good at energy storage. If we zip back to these carbohydrates, there are a lot of covalent bonds here in these monosaccharides, in these disaccharides, these polysaccharides. There's a lot of energy you can get out of this, but there's also a lot of wasted space. This has formed a ring structure, which is not the most efficient thing, just space-wise. This is a great way to store energy, but it's not the most dense way to store energy. If you want to talk about dense energy storage, just look at this brick of carbon here. Fats and oils are way more efficient energy storage compared to carbohydrates, but it's a little more difficult to fully break this thing down. So this type of energy storage tends to be long-term storage. You'll do your carbohydrates on an hour-by-hour basis, but in a longer term, if you want deeper storage of energy that's a little more difficult to access, that's what these fats and oils are used for. More efficient, long-term storage compared to carbohydrates. And yeah, a side note about nutrition, if you want just a sense of how much more densely packed this is compared to carbohydrates, if you look at a gram of carbohydrates, that's, I think, about 4 calories worth of energy in a gram of carbohydrates. A gram of fat is going to have about 9 calories, if I remember correctly. So the point is, it's way more densely packed. So yeah, seems redundant. It's more energy storage. This is your long term. Okay, enough about fats. How about lipid number 2, waxes? These are a lot simpler. It's not 3 fatty acids on a glycerol backbone. Waxes are just a single long fatty acid. Let's take a look at these things, and yep, there's paraffin. The carbons and hydrogens are not even labeled here, but yeah, each one of these things is a carbon, 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 carbon. Yeah, long chain. Sometimes you have little branch points, isoparaffin, cycloparaffin, there's a little ring here, but yeah, it's a single long fatty acid. What are these things used for? Well, they're not really used for energy storage, but as we mentioned, when we first started introducing lipids, these things are hydrophobic. The fact that lipids of all types are hydrophobic means waxes can be used for waterproofing. So plants will, use waxes in their cuticle to waterproof themselves. Lots of animals will use waxes, in their fur or on their feathers to waterproof themselves. So yeah, very, very different kind of function, not energy storage. Waxes are used by plants and some animals to help repel water. Again, these things are hydrophobic. And that's all I have for waxes. This is a pretty simple one. Next, we have something called phospholipids. So a phospholipid consists of 2 fatty acids and 1 phosphate group. That's why they're called phospho. on a glycerol backbone. So this is the second time we've seen a glycerol backbone. In fact, phospholipids are very similar to triglycerides, except instead of having a glycerol backbone and 3 fatty acids, in the phospholipid we have a glycerol backbone and 2 fatty acids. They could be saturated, they could be unsaturated, whatever. So instead of that 3rd fatty acid tail, phospholipids have a phosphate group. That's one of our functional groups. And it has a P here, part of our sponge. This is the phosphorus element. Now, these are very hydrophobic. We talked about these fatty acid tails. There's no charge in here at all. These really don't want anything to do with water. But the phosphate group, this functional group here, if you look real closely at it, there's a little minus sign right here. The phosphate functional group is charged with a full, not even partial, a full negative charge. So when you have a phospholipid, that means part of it is hydrophobic, but part of it, the phosphate group here, is very hydrophilic. It loves water. It wants to get surrounded by the partial positive charges that we find in H2O. This creates a very interesting type of molecule that is both of these things. Half of it's hydrophobic, half of it's hydrophilic. So fatty acids, as part of this phospholipid, are hydrophobic. The phosphate group is hydrophilic with a negative charge. Now, this is a detailed way of drawing a phospholipid, showing the carbons and the phosphorus. A lot of times these are cartoonized into something like this. Here's the polar or hydrophilic part of the phospholipid. These are the non-polar or hydrophobic tails of the phospholipid. And when you take a bunch of these and put them together in water, they will arrange themselves in such a way that the heads are touching water and the tails are hanging out with each other. Because again, these tails don't want to touch water, but the heads do. If we want to look at this in another way, here is what is called a lipid bilayer sphere. We're looking at a cross-section here, in case you're wondering, hey, these tails would be touching the water and their surroundings. We're looking at a cross-section, and this is fully three-dimensional. None of these tails would be touching water at all. They would just be hanging out with each other, which is exactly what they want. The heads on the outside are touching water, and the heads on the inside, well, they're touching water as well, because there's water inside of this lipid bilayer sphere. This seems like a weird, funky-looking structure. This is actually essentially a cell. Our cells are lipid bilayer spheres. So the whole basis of a cell is this bilayer of lipids, of phospholipids, with stuff on the inside, water and organelles and other molecules or whatever, and stuff on the outside. So phospholipids in specific are very important components of the cell membrane, which we'll talk more about in a later chapter. But yeah, to introduce these now, the properties they have, phospholipids form the cell membrane. Okay, one more type of lipid. Number 4, steroids. So this is a really weird one, both in structure and in function. So for the first time, we don't see a big, long, fatty acid tail that could be saturated or unsaturated. Steroids instead are 4 fused rings. And again, I don't expect you to memorize or draw any of these structures, but yeah, these are some steroids: cortisol, estradiol, testosterone, and yeah, they all have 4 carbon rings that have fused to one another. These are primarily hydrophobic. A few OHs here, so there's a little bit of water loving, but on the whole, all of these ring things are hydrophobic. I said that about lipids in general, that lipids generally are hydrophobic. And yeah, they're kind of defined based on their structure of being composed of fused rings. So that's their structure. What's their function? Well, there are a couple of different functions of steroids. One of these is cholesterol, which, if we zoom in on cholesterol. Yep, 4 fused rings, that's a cholesterol. Cholesterol is actually part of the cell membrane. So yeah, going back to this again, the cell membrane is mostly phospholipids, but when we look at it more closely in the next chapter, I believe, we're going to see cholesterol in there as well. So some steroids like cholesterol just help stabilize the cell membrane, part of this cell membrane structure. Something that we're probably more familiar with, though, are steroids that act as signaling molecules. So there are many other steroids, like testosterone, for example, that function as hormones. So we'll see hormones later in this chapter as well. A hormone, to read from the key terms here, is a chemical signaling molecule that acts to control or regulate specific physiological processes. In other words, a hormone is a messenger. So many of these steroids, like testosterone and others, their whole job has nothing to do with energy or structural integrity. The whole job of some of these steroids is to just send a message. And again, it's kind of a weird job for a lipid, but yeah, these are a type of lipid based on their hydrophobic characteristics. So steroids, including testosterone, hormone, is our 4th type of lipid. Okay, we have more biological macromolecules to get to, but this is typically where I end things for this recorded lecture, so we'll do the rest of this chapter in the next one. This is the end of recorded lecture 1-3.